All around us, and even inside of us, there's this entire invisible world just buzzing with microscopic life. But have you ever stopped to think about how a single celled organism actually navigates its incredibly complex environment? Well, it turns out they talk. They have this really sophisticated communication network that lets them sense what's happening, talk to each other, and make these critical life or death decisions. So today, we're going to dive deep into the fascinating world of microbial signaling.
So here's what we've got on tap. We're going to start with the big question. Why do microbes even need to talk in the first place? Then we'll get into the nuts and bolts of the classic bacterial relay, the two-component system. After that, we'll see how bacteria literally count themselves using quorum sensing. And then we'll jump into the more complex world of a eukaryotic pathogen, the malaria parasite, to see how things change. And finally, we'll tie it all together with a summary.
All right, let's jump right in. Before we get into all the molecular details, we really need to understand the why. Why is all this signaling machinery necessary?
So, really, why this constant vigilance? Well, for an organism that's just a single cell, its immediate environment isn't just some passive backdrop. It is everything. It's where the food is, it's where the dangers are, it's where it finds a home. Survival is a constant moment-to-moment challenge that completely depends on being able to sense what's out there and respond fast.
I think this image is a perfect analogy. The crossing guard, she's like the microbe's sensor. The stop sign is the environmental signal, the cue. And the action, the kids crossing the street safely, that's the microbe's response. It's exactly the same principle. A microbe gets a signal, maybe a shift in pH or the scent of sugar, and that triggers a very specific, appropriate action that helps it survive. It's all about cause and effect, stimulus and response. So, what exactly are they keeping tabs on? Well, pretty much everything. They're constantly monitoring physical conditions like temperature, pH, oxygen levels. They're on the lookout for food, of course, but also for things that could kill them. And this constant surveillance is what makes them so incredibly adaptable. I mean, how else could a bacterium go from living in a puddle to surviving in the human gut? But here's the key thing to remember. The ultimate goal of all this sensing, all this information processing, it almost always boils down to one thing: changing which genes get transcribed. Okay, so how do bacteria actually do that? How do they connect an outside signal to a change in their DNA expression? Let's get into our first big mechanism, the two-component system. This is probably the most fundamental and widespread signaling pathway in the prokaryotic world. It is the absolute workhorse of bacterial sensation. As the name suggests, you've got two main players. First, there's the sensor kinase. Think of this as the cell's antenna. It sits right in the cell membrane with a piece of it sticking out, just feeling what's going on in the outside world. Then, inside the cell, you have the response regulator. This guy is the internal messenger. As soon as the antenna picks something up, the response regulator is the one who takes that message and delivers it right to the cell's command center, the DNA. And this is how that message gets passed along. It's basically a molecular game of hot potato called a phosphorelay. So, step 1, the sensor kinase detects a signal. In response, step 2, it does something called autophosphorylation. It grabs an ATP molecule and sticks one of its phosphate groups onto itself. Step 3 is the handoff. That phosphate is immediately passed to the response regulator. And that's the trigger. Step 4, once the regulator has that phosphate, it's activated. It can now go bind to the DNA and flip the switch on specific genes. Let's make this real with a classic example, the AMPAR/NVZ system in E. coli. This system is all about osmoregulation, basically making sure the cell's internal salt and water balance is just right. In this pair, NV is the sensor kinase that detects salt levels, and AMPAR is the response regulator that controls what happens next. And what they control is the size of the pores in the cell's outer membrane. And the logic here is just really elegant. When the cell is hanging out in a low-salt environment, it makes a porin called AMP-F, which has a pretty big opening. That's great for letting in lots of nutrients. But if the cell finds itself in a high-salt environment, that big porus suddenly becomes a liability. So the cell switches gears and starts making AMP-C instead, a porin with a much smaller hole, which helps protect the cell by restricting what comes in. So how does the cell decide which one to make? It's all right here in this table, and it comes down to the concentration of the activated response regulator, AMP-RP. Check it out. In a low-salt environment, you get low levels of AMP-RP. That low concentration is just enough to activate the gene for the big AMP-F pore, but the AMP-C gene isn't touched. Now crank up the salt. The NVZ sensor goes into overdrive, and you get a high concentration of OMP-RP. This high level does two things at once. It switches on the gene for the small OMP-C pore, and at the same time, it binds to a different site on the OMP-F gene and actively shuts it down. It's a beautiful, precise, dose-dependent response.
Okay, so we've seen how a single bacterium can listen to its physical surroundings. But bacteria are rarely alone. So what happens when they want to act together as a coordinated group? Well, that brings us to our next big idea, quorum sensing.
Quorum sensing is basically bacterial democracy, or maybe more like a census. There are certain things bacteria do, like launching an attack to cause disease or building a tough biofilm, that are pretty useless if you're doing them alone. They only work if a whole bunch of bacteria do them at the same time. Quorum sensing lets bacteria hold their fire, to wait until they've confirmed there are enough of them around to actually make a difference.
So how does it work? It's all based on a small signal molecule called an autoinducer. Every single bacterium is constantly making and releasing a tiny amount of this molecule. When there are only a few cells, the signal just floats away. But when the population gets dense, that signal starts to build up in the environment. Once it hits a critical concentration, it starts flooding back into the cells, where it binds to an activator protein and flips the switch for all those group behaviors. And the classic textbook example of this is the incredible relationship between the bacterium Allevia librio fisheri and the Hawaiian bobtail squid. These bacteria live inside a special light organ in the squid. When they're free-living or at low numbers, they don't glow. But once they multiply to a crazy high density inside that organ, their quorum sensing system fires up and they all switch on their lights at once. The squid gets to use this light as camouflage, and in return, the bacteria get a safe, cushy place to live. And these are the genes that make the magic happen. It's all part of the Lux operon. You've got the Lux-I protein, that's the factory that makes the autoinducer signal. When the signal concentration gets high enough, it binds to Lux-R, which is the activator protein. This activated Lux-R then latches onto the DNA and turns on the whole rest of the operon. Lux-A and B, which make the enzyme that actually produces the light, and Lux-C, D, and E, which make the fuel for the reaction. It's a perfect self-contained system. Right, we've spent a good bit of time in the bacterial world. Now let's completely switch gears and look at a eukaryotic microbe, the malaria parasite. And as you're about to see, the signaling pathways here get a whole lot more complicated.
So here's the biological puzzle we need to solve. The malaria parasite spends part of its life just circulating in human blood, reproducing asexually. But to complete its life cycle, it has to get picked up by a mosquito. And the second it enters the mosquito's gut, it has to immediately switch over to sexual reproduction, a process called gametogenesis. How does it know? How does it realize it's left a human and entered a mosquito? What's the trigger?
Well, finding that trigger was a real scientific detective story. For decades, researchers knew something in the mosquito gut was kicking this process off. They gave it various names like MEAF and GAF. But it wasn't until modern high-res mass spectroscopy came along that they finally nailed it. The molecule is xanthurenic acid, a breakdown product of tryptophan. And here's the crucial part. It's found in the mosquito, but it's not found in human blood. That's the signal.
And just look at the cascade that one little molecule sets off. We are not in the simple two-component world anymore. Xanthurenic acid binds its receptor. That triggers the production of a second messenger, cGMP, which amplifies the signal inside the cell. The cGMP then activates the first protein kinase, PKG. This kinase then triggers the next big event, a massive release of calcium, our second messenger. That flood of calcium then activates a second kinase, CDPK4, and that is the final switch that gets gametogenesis going. It's not a simple relay, it's a full-blown amplification cascade. Each step magnifies the signal, turning a tiny whisper from the environment into an undeniable all-or-nothing command inside the cell.
Whew. Okay, we have covered a ton of ground, from those simple bacterial phosphorylase to these complex eukaryotic cascades. So let's just take a moment to pull it all together and recap the main principles. So what are the big takeaways here? First, microbes are constantly sensing their world. In bacteria, this is often handled by these beautifully simple and direct two-component systems. They also have ways to talk to each other and to act as a group, which we see in quorum sensing. But as we move from prokaryotes to eukaryotes, like the malaria parasite, the game changes. Those simple relays evolve into much more complex, multi-step cascades that use second messengers and multiple kinases to amplify signals and create these really robust, fine-tuned responses.
And that really brings us to a final, pretty important thought. We've been looking at the molecular nitty-gritty of how these microbes communicate. But if you step back and think about the bigger picture, if a pathogen needs quorum sensing to launch an infection, or if the malaria parasite absolutely requires xanthorrhinic acid to reproduce, what if we could intercept those signals? What if we could jam their communications? You see, understanding this secret language doesn't just tell us about how microbes live. It could give us brand new ways to fight infectious diseases. Thanks for joining me.